Hello everyone, my name is Gantt Meredith and today I'm covering an exam 2 topic known as microscopy. So let's go ahead and get started. In a general sense, what is microscopy? Well, it's simply the use of a microscope to observe a material or a substance. We have an image below of a microscope, and the rounded objects are different lenses of magnification for you to use to observe the sample given. Now, there are many types of microscopy. Today I'm going to focus on the 3 types of microscopy that are relevant to this course. However, at the end I will touch on a very interesting and very exciting form of microscopy that is used in the field of biology. And me being a biomedical engineering major, I actually use this in one of my classes, cellular engineering laboratory, but we'll get there at the end. So for right now, let's focus on types of microscopy that are relevant to this course. We have optical microscopy, electron microscopy, and scanning probe microscopy. And we gave a little scale here just so you can see what 1 inch, 1 centimeter would look like with these images. But as you can see, we'll discuss this more in depth in a second, but optical microscopy gives a fairly rough image. Electron gets a little bit closer, we're able to see more of the actual structures and orientations and shapes present. And then if you look at the scanning probe microscopy, well, we get pretty detailed. It looks almost like a very spongy, very porous, and we're able to see the different spaces between the atoms present in a compound or material. Let's break those down one by one. Let's look at scanning probe microscopy. Now, it is what it sounds like. There is a physical probe that is going, pardon me. A physical probe is going to scan the entire surface or a selected surface of a material. And now the way that works is we have a field going from a piezo scanner or piezoelectric. If you've taken baby circuits here at UF, we use piezoelectric buzzers in our circuits. And so it makes a field with the probe. The probe then scans over the sample material, and there is a feedback signal that the probe can then interpret. This type of microscopy is actually fairly recent. It was only formalized and regulated in 1981. And you can actually get a relatively high atomic resolution from this. If we look back up here, you can get a pretty high resolution from that. Now if we look at electron microscopy, how this is different is there's actually a stream of electrons that travel down the microscope. You can see we have a high voltage coming in, an electron gun, and then the electrons are condensed and focused to an electron beam. Now at the bottom we have a fluorescent screen and a camera, and these harness the electrons and they give us an image that we can then interpret and use for analysis. It's important to note that electron microscopy is extremely precise and accurate, and if you look up here, electron microscopy and scanning probe microscopy, while they're both very accurate, they give us different looks at compounds. So it can be very interesting with the different compounds, crystal structures, lattice structures that we're given to observe them under different lenses. Optical, electron, and scanning probe microscopy can all give very different representations of what a compound or substance actually looks like. The last type of microscopy relevant to this course is going to be optical microscopy. We know that optical has to do with they both have to do with the eyes and vision, and so you can look up here. You actually will put your eyes on the eyepieces and look directly at the material or substance. You can adjust the magnification here. Normally, especially in my cellular engineering lab, the optical microscopes that we use range in magnification from 5 times to 20 times, but 20 times magnification to still get somewhat precise, but the images are not nearly as detailed as electron and scanning. And if we go back up to the beginning, that we can see a representation of how there's a lack of detail in optical microscopy. Now, as engineers, we are going to come into contact with other majors, other disciplines. So especially in materials, one area where we might use something called fluorescent microscopy is in biomaterials. So if we look here, these are called HeLa cells. HeLa cells, H-E-L-A, is short for Henrietta Lacks. She was a woman who had cervical cancer, and her cells actually wouldn't die, so they were able to be studied for an extensive amount of time and replicate. We're still using her cells today, and they're actually the most well-researched cells used for cell passaging, cell culture, and many different research techniques. But fluorescent microscopy is very, very cool because we can take staining solutions and we can stain these. We can stain the HeLa cells, we can stain their nuclei, we can stain different structures and compounds within them, and then we can use fluorescent light or a fluorescent microscope to absorb, to view the bioluminescence or the biofluorescence present in the HeLa cells. So if you look on the right, you'll see that the HeLa cells are green. We used, in cellular engineering lab, we used a stain known as the Hocht stain, and the Hocht stain actually does give you green fluorescence if the cells are alive. Now the really cool part about fluorescent microscopy is that you can overlap images. So you see the green here, but we also have blue, and we also have red. And these are different stains that can be measuring different things, such as fibers, strands, dead parts of cells, active parts of cells. The list goes on. And this is a very important aspect of biomaterials, especially because if you're working with a drug delivery scaffold, for example, you want to be sure that your cells are able to live, that the drugs are delivered accurately, and the drugs are delivered over time in an appropriate manner. And fluorescent microscopy is just one way to do this. While this might not necessarily be exam material, it's important for us to realize that microscopy is a very, very huge topic, and there are many applications. But for the purposes of the exam, just focus on these three. I hope this was informative, and I hope that you have a better understanding of microscopy now. Thank you.